Guys, it's Mrs. Strasden. We're going to go over some fill in the blank notes that you can do on your copy. The title of the notes is DNA Replication. Replication just means the DNA is going to copy itself. So this is the learning target 5.1, describing the structure and function. And we're focusing again today on DNA. We'll get to RNA next week. So DNA is really long, we know this. So we have some vocabulary terms coming up. In order to fit into the cell, which that's one of the blanks you have to fill in, it coils around proteins. And these proteins are called histones. So it's going to wrap itself around a protein. It's like a rope coiling around a barrel, is what that's like. These coiled strands of DNA, when they're all coiled up, are called chromatin. And then these chromatin pack together even tighter. I mean, we got to fit all this into the cell. So they pack even tighter together, and we call those chromosomes, which you may have heard chromosomes before. So chromosomes are just tightly packed DNA so that it'll fit into the cell. We humans have 46 chromosomes, and they kind of match up 23. We call them 23 pairs. And comparing, carrots only have 18 chromosomes. So just a comparison. You have your word bank for when you go to fill in your copy. There's a picture of our 23 chromosomes. And you can see 1, 2, 3. So you hear sometimes of mutations or different syndromes like Down syndrome. Down syndrome has to do with a specific number of the chromosomes. So like number 22, there's an issue with it, or 19. And so we'll talk about, when we talk about mutations, we'll talk about some of those mutations that might happen. So when a new cell is made, it needs a copy of that DNA. And so DNA will replicate or copy itself to give to the new cell. So when it does this, the DNA molecule splits. So it's all tightly wound. It's going to come loose, and it's going to split into 2 loose strands. We're going to see a picture of this. And then new free nucleotides will line up accurately on the other side. And then you get 2 new complementary strands of DNA made. So we'll look at it in a picture, because I kind of said that in a mouthful. But when you see the picture, you'll kind of see what we're talking about. Those nucleotides line up following the rules of base pairing. Those rules, remember, adenine goes with thymine, and cytosine goes with guanine. So if we look at an example here, we see a single strand of DNA has T, A, G, A, C, T, A. So that's thymine, adenine, guanine, adenine, cytosine, thymine, and adenine. So the other side that's going to line up and match up with it, well, since this is T for thymine, A is going to line up with it, adenine. And since up here it was A, adenine, T or thymine is going to line up with that A. And since G is guanine, cytosine is going to match up with that G. And so we just kind of match these up. Adenine goes with thymine down here. Cytosine goes with guanine down here. Thymine goes with adenine right there. And then adenine goes with thymine. So we just kind of match up according to that base pairing. So you're going to get to try it right here. They give you G, G, A, C, T, T, A. Well, your answer would be C to go with the G, another C to go with that G, an A to go with T, and so forth. So if we look at the picture of DNA copying itself, this is pretty complicated. I don't want you to get stressed out about all the terminology. But I'm going to add a little video to this because my 5 minutes is up. And we'll talk through this